Welcome to The Explainer. Today, we're decoding a truly fascinating intersection of disciplines. We're looking at exactly where the messy, organic world of biology collides head-on with big data and computer science. It's a field that has completely revolutionized how we understand life itself. So we're going to break down exactly how it works using some brilliant insights from Chapter 9, Introduction to Bioinformatics. Okay, let's dive into this. Here's our roadmap for navigating the chapter. First, the biological data explosion. 2, the pioneer of bioinformatics. 3, math and stats in biology. 4, generating and analyzing data. And finally, number 5, decoding the genome. Part 1, the biological data explosion. To really grasp bioinformatics, you first gonna have to understand the massive, honestly almost overwhelming problem that modern biologists suddenly found themselves facing. Think about how biology used to be done, right? Biologists could generate and analyze their experimental data using simple notebooks or, maybe standard Excel sheets. But the advent of powerful new technologies like high-throughput DNA sequencers and advanced imaging systems changed absolutely everything. Suddenly, biologists weren't just collecting data, they were hit by a total data tsunami. The sheer volume of information being generated was just way beyond human scope to manage manually. You literally couldn't just write this stuff down anymore. And this brilliantly illustrates the core issue. Biological data isn't just a simple list of numbers. It forms a highly complex web of networks and interactions, much like these molecular structures. To retrieve meaningful, vital information about proteins, genes, and cellular mechanisms from this tangled web, biologists had to turn to computational algorithms and massive databases. They essentially needed computers to see the patterns that the human eye just never could. Part 2, the pioneer of bioinformatics. Now, computers obviously don't program themselves. Behind this computational revolution were some incredibly visionary human beings who saw the data problem coming from a mile away and started building the solutions. One of those absolute pioneers is Margaret Oakley Dayhoff. She was an American physical chemist who dedicated her entire career to applying mathematics and computational methods to biochemistry. And what's really amazing is that she did this well before modern genomes were even sequenced. Back in 1965, she published the Atlas of Protein Sequence and Structure. It was a comprehensive, open-source collection of protein sequences that fundamentally changed the game. But her genius didn't stop at just collecting the data. She actually knew how to optimize it for the technology of the time. Dayhoff developed the one-letter codes for amino acids. Why? Well, in an attempt to reduce the size of data files in early memory-limited computer applications. This ingenious, space-saving solution became the foundational model for the massive sequence databases that were developed later on. She essentially created the language that allowed biology and computers to speak to one another efficiently. Part 3, math and stats in biology. All right, so now that we have the computers and the data, we really have to talk about trust. How do we know the patterns that computers find are actually real? A strict mathematical foundation is absolutely required here. For example, let's say we want to understand the association between heart rate and systolic blood pressure in a group of patients. A simple visual estimation isn't going to cut it. We need stats. We use correlation to quantify exactly how those two variables are connected, but we use regression, like drawing a regression line through our data points, to define a statistical relationship where a change in one variable directly affects the other. Understanding these distinct tools is just absolutely crucial for extracting actual biological meaning from raw numbers. And as you probably know, in statistics, you always have to be your own biggest skeptic. This is where the null hypothesis comes into play. It's basically the baseline statement assuming that there is absolutely no relationship between the two phenomena you're measuring. In bioinformatics, we have to constantly test our massive data sets against the null hypothesis to ensure that our experimental results are genuine, and not just fake anomalies or random statistical noise. So the crucial point here is understanding your p-value. The p-value tells you the probability of finding your observed results if the null hypothesis were actually true. If your p-value is low, your result has statistical significance, meaning it's very unlikely to have occurred by mere chance. This mathematical rigor is our absolute primary defense against false positives when we're sifting through billions of data points. Part 4, generating and analyzing data. With our statistical foundations firmly in place, let's open up the modern biologist's toolbox. The sheer volume of our data comes from incredibly fast, high-throughput technologies. We use RT-PCR to quickly detect RNA expression. We rely on HPLC to separate and quantify metabolites. And mass spectrometry determines the mass and fragmentation of molecules. These are literally the modern engines pumping out the vast raw data formats that bioinformaticians have to analyze every single day. Now, what's really interesting about this is how visually striking the data can be. This is a chromatogram from a mass spectrometry reading. It really shows how modern biology has become a highly visual, data-driven science. All those peaks and valleys represent relative abundance and mass spectrums. To the trained eye, and of course the trained algorithm, this image holds incredible biological insights.
To process visuals and data like that, we rely on common analytical workflows. We use tools like BLAST for homology searches to find similar sequences. We use ClueStall or MAFT for sequence alignments. And we use powerful statistical packages like R, SPSS, or SAS. But here's the key takeaway for this part. Just knowing how to run the software isn't enough. A biologist must genuinely understand the underlying logic and assumptions of these tools to ask the relevant questions and properly interpret the biological results.
Part 5. Decoding the genome. Okay, so we've gathered the massive data, we've applied the software, and we've done the math. Now what? What do we actually do once we've sequenced a massive genome? Turning raw sequencing data into functional biological meaning follows a very logical progression. Step 1 is sequence assembly, where we combine millions of tiny sequenced fragments, called reads, into longer contiguous stretches known as contigs. Step 2 is genome annotation. This is where we act like molecular detectives, scanning that assembled genome to identify the coding parts of genes, the non-coding elements, and the repeats. And finally, step 3 is gene ontology, where we take those identified gene products and unambiguously describe their associated biological processes and molecular functions. Let's move along and see how this actually builds our understanding of life's history. During that annotation step, we often find repeat elements, which make up a really significant chunk of eukaryotic genomes. These repeats, things like transposons and satellites, they aren't just junk. They actually provide vital diversity to individual genomes and play a highly significant role in evolutionary history. By analyzing the data, we are quite literally unearthing the hidden evolutionary history, coded right into our DNA. And that brings us right to the frontier of this science. We started with Margaret Dayhoff inventing a one-letter code just to save precious computer memory. Today, the biological data explosion is only accelerating, producing unprecedented volumes of complex information. So the ultimate question we want to leave you with today is this. As our data sets grow infinitely larger and more complex, how will artificial intelligence step in to take genome informatics to the entire next level? The algorithms of tomorrow are poised to uncover biological secrets we probably can't even fathom today. Thanks for joining us for this explainer.